So our next speaker is David Loehlin, who's going to talk to us about non-additive expression of tandem gene duplicates.
Hi, everybody. My name is David Loehlin. I'm a postdoc in the Sean Carroll lab, and I study differences between different species of flies, and I'm going to talk about tandem gene duplication. So a lot of biologists are interested in tandem duplication or duplication in general as a source of novelty. So once you have two copies of a gene, you have a functional redundancy, which then can evolve new functions or new expression patterns. Very important in evolution and in expansion of gene families and things like that. Something which I think we know comparatively less about, although I've been hearing interesting things in this meeting, is how this happens in the first place. So how does a new duplication arise? How does it spread in the population and stick around for long enough for diversification to happen? So what I'm going to talk about are the initial phenotypic consequences of tandem duplication itself. In general, so I came to study duplication almost by accident. I'm interested in how adaptive evolution shapes genes, in particular in a quantitative way. And I like evolution because even in a simple adaptive model, the optimal value of a trait changes, and the way that a species or population compensates is by accumulating a bunch of beneficial mutations, pushing it to the new optimum. Now, if you think about gene structure, if you're interested in gene structure, it would be really cool to know what these beneficial function-altering mutations are, where they occur in a gene, and if they tell us something about which parts of the gene evolution likes to use. So what I study as a model for this is flies adapting to alcohol, and in particular the alcohol dehydrogenase gene, which is very important for how flies process alcohol. It's co-evolved in a bunch of species which have higher or lower ethanol tolerances. For example, Drosophila virilis you can now collect it in breweries, has a very high alcohol tolerance, whereas sibling species like Drosophila americana, they seem to like willow sap. They have very low alcohol tolerance. And if you grind up the flies and just measure the activity of their ADH enzymes, you can see that there's a huge difference. So virilis has about 6-fold higher ADH enzyme activity than americana, which is, as a geneticist, this is a really nice trait to study. I want to find out how this difference occurred. Now, it's been known for a while that virilis is a tandem duplication of ADH, and I figured naively that a tandem duplication is going to give you twice as much enzyme, but the species difference is 6-fold, so I figured there would be other coding changes, regulatory changes, possibly trans changes, which are part of this difference. So to study this, I cloned these genes and was attempting to pick them apart, and in the process of doing that, I ended up, again, sort of by accident, doing an experiment where I actually just directly compared the effect of the tandem duplication itself. So I'm going to show you this experiment. This is a direct test of the hypothesis that the tandem duplicate gives you 2-fold higher enzyme activity than the single copy. So I have a tandem duplication of ADH from virilis. I should mention that the transcript is about 3 kb and the whole block is 7 kb, and this includes all the known regulatory elements. In this experiment, we have a tandem duplicate and a single copy where there are no other SNPs among any of these, so it's really just comparing the effects of duplicate to single copy. We use the Phi-C31 at-P system to put both trans genes at the same location in a controlled genetic background. And the results of this experiment were the surprise. So the y-axis is ADH enzyme activity, and there's a box plot with a 95% confidence interval for the same set of data. And what you can see is the single copy is low, the duplicate is high. The dashed line is twice the single copy, and what you can see is that the duplicate activity is not twice the single copy, it's significantly greater. It's actually 2.7-fold higher than the single copy, suggesting that the tandem duplicate is not giving us 2-fold, it's giving us some excess. So I thought, hey, that's pretty cool if that's true. It might suggest that there's a potential general property of tandem duplicates. And tandem duplicates you can get in almost any organism. this could be a big deal. But I had to convince myself first that this was not just a scaling issue. You know, there could be something nonlinear, non-additive about this assay. And also there's issues of copy number per cell, which we heard a little bit about in the previous talk, for example. So to control for copy number per cell and really get a sense of, is it really the duplicate structure that matters, I did a cross and compared duplicate heterozygotes, so 2 copies over an uninserted chromosome, 2 copies per cell, to singleton homozygotes. Both of these flies are going to have 2 copies per cell, so we can actually look at the effects of tandem configuration versus 2 copies in a trans configuration. And I did a cross, so you end up with 4 genotypes actually, but the important thing are the 2 in the middle. And you can see that the duplicate heterozygotes do have a significantly greater enzyme activity than the singleton homozygotes. Same copy per cell, but the duplicate configuration is giving you the excess. And what that suggests is it's the structure of this gene that's giving you the excess. And so it's something at the DNA level. And that suggested to me that we would not just see this at the protein level, the level of enzyme activity, but also at the transcriptional level. So I did quantitative PCR. And unsurprisingly, the tandem duplicates have greater than 2-fold expression at the RNA level of the single copy. So it does seem like it's both transcriptional and at the protein level. So it would be cool to know if this is a general phenomenon. We have evidence from one gene, and I thought, well, maybe this is a peculiarity of ADH. So to test that, I wanted to duplicate another kind of gene and see if you get 2-fold or less than 2-fold or greater than 2-fold. And the gene I selected to do this is we decided to pick something that's very different from ADH, but still lets us do these enzyme assays. So we picked a reporter construct. So this is the beta-galactosidase enzyme from E. coli, driven by the vestigial quadrant enhancer, which is active in the wing imaginal disc. This is just something we had in the lab for developmental studies. So I took this, made a single copy and a tandem duplicate copy, and inserted single and double at 7 different at-p sites on the autosomes. This different locations allows me to control for any possible position effects, which might be affecting expression differentially. And so here's the results of this experiment. This is the activity of the singleton. This is the activity of the duplicate. So like this point, the singleton is giving you about 60. The duplicate's giving you about 130. And the important thing is the dashed line is what a 2-fold difference between singleton and duplicate is. And you can see that all the points are above the line. I will tell you that they're all statistically significantly greater than 2-fold. But what's interesting is a couple of these, like the one labeled 4 or the one labeled 2 up here, the duplicate isn't just a little bit more than 2. This one, the duplicate is 5-fold higher expression than the singleton, suggesting that there are position effects, and you can see position effects even if you just look at the singleton axis and say, okay, well, this one's less than this one's less than this one. But there's position effects both for activity of a single copy and the difference between singleton and duplicates. So there's something going on that's interesting with respect to how the duplicates are interacting with each other and with adjacent sequences. So this is kind of a phenomenological thing. This is the chromatin and regulation section, so I'm sure a lot of you are thinking about the mechanisms. And basically, I've told you everything that I know about this right now. So I'd be interesting if you have some thoughts about this, how this works. You know, there are chromatin hypotheses, there are DNA folding hypotheses. The one that I like, and this is speculation, that I think brings this stuff together comes from the transcription factory theory. And that's the idea that the polymerases and transcription factors tend to occur in big clumps, so that if a gene is being transcribed or even regulatory elements are bound, adjacent DNA is also right near the transcription apparatus. So if you have a tandem duplication, you have 2 genes with similar regulatory elements. Initiation or transcription of one is going to bring the other one right in close. So almost deterministically, you might expect that tandem duplicates would have this excess of transcription. And then additionally, what's going on out here, if these are similar or different binding sites, could affect the degree to which this happens. And there may be some ways to test that, but if this is something that you work on, I'd be interested in talking to you. And yeah, I should just say that's speculation. So the last point I have to make is, if you've looked at the literature, there have been some interesting papers on the long-term trend of what happens to tandem duplicates. And for example, Land and Pritchard compared different groups of mammals, and what you see is over the long term, natural selection pushes expression of tandem duplicates down to the single copy level. So not twofold, but onefold. What I've shown you is that, at least in this case, the initial difference is not just twofold, but it's significantly greater than twofold, so it's going in the opposite direction. This potentially could explain why we see very few tandem duplicates in populations and why they tend to occur at low frequencies. But of course, I think we need to do a lot more investigation, see if this holds for a lot of different kinds of genes, see if this occurs in other organisms, and just find out more about this process, and if this greater than twofold rule holds, or if there's some different rule that happens in other contexts. But it could be fun and interesting. And thank you very much. Yeah.
So with respect to the transcription factory hypothesis, wouldn't you expect to see then greater expression if you just put one copy of the gene, say, downstream of any highly expressed gene?
Yeah, I think you would expect that.
Is it about the duplication or is it about the transcriptional environment? And I don't know if there's any examples or any way to systematically do that with the existing genomic architecture if you would have to engineer something, but that might be interesting to look at.
Yeah, I mean, one prediction from that is that if you just add in addition the promoter, for example, or the regulatory elements, it should also have that effect. So I think that'd be interesting. I think the transcription factory theory is sort of new, and it would be nice to accumulate some evidence, look at neighboring genes. Certainly, genome-wide, it's been observed that genes which are adjacent to each other tend to have genes which are have similar expression patterns tend to have higher expression levels or something like that. So.
Thank you. Nice work. Two very quick questions. One is, it looked like at the transcription level you actually had way more than 2.7-fold increase. And then I think that one of the most simple explanations might be that you're just doubling up on regulatory regions that can act any which way. Have you tried just mutating the second copy of the actual gene or moving an insulator in between?
Those are good suggestions. I've thought of them, and they're a good idea to do. Building tandem duplicates is slightly more irritating than building, little regulatory element constructs, but those are experiments I want to do. And yes, you're right. At the transcription level, the quantitative PCR shows greater expression than I see at the enzyme level. I don't know if the scaling is right, but it could just be that you are saturating translation of these very highly expressed things. ADH is, I'm told, like one of the highest expressed genes.
Can I suggest that you put an inverted repeat on your list?
Yeah, that's a great idea. There is the issue that an inverted tandem repeat will form a hairpin, but I would like to do some form of experiment there.
Do you know if the transcript coming from the tandem repeat contains both in a sort of polycistronic type form?
Yeah, that's a good question. This could be transcriptional read-through. There's a Northern blot and a Viralis paper from the 90s which only shows the correct size transcript. And then in the LAC-Z experiments, there's an SV40 terminator, which I'm told is strong, but I haven't actually done those measurements.
Thanks.
Hi, I may have missed this, but since you're looking at variation because of the point where you're inserting things, so different ATP sites give you different variation, would you expect to see the same differences if you used a plasmid-based assay? So if you had a plasmid with tandem repeats that you transfect into S2 cells, versus, if you had just single copy. I don't know how you would control for transfection efficiency in that case, but if it was a recruitment of transcriptional machinery effect, then you would expect to see it in a plasmid-based assay as well.
I haven't ever thought of that. That's a good suggestion. That's interesting. I mean, there's no chromatin or something, so
Yeah.
yeah,
So.
good idea. I think we're out of time.
Yeah, I think we've probably got time for one more quick question.
Well, it's mostly a comment. We should talk afterwards. So the replication-dependent histone locus in Drosophila is a tandem array of 100 copies. And it's associated with a nuclear body called the histone locus body, which is effectively an RNA biosynthetic machine, analogous to what you're talking about. So we have evidence that if you have 12 tandem duplications in cis, you get more function than if you do 6 over 6 in trans. So it's very similar to what you're talking about. So
Oh,
we
cool.
should chat afterwards.
Yeah, that would be great.
Okay, at that point, we probably better move on. Thank you again, David.